A very good afternoon to all of you, and welcome to the first lecture in this series that's being organized by the Indian Pollinator Initiative. I welcome today's speaker, Alyssa Stewart, and thank you all for your response. We've had an overwhelming response to this seminar series, and we look forward to participation in the coming weeks as well. So we have a lineup of a good lineup of speakers who work in the field of pollination research, the first of which is today. And to tell you very briefly about how this thing came about. So the idea was mooted by Axel Brockman, who's here. He's from NCBS. And a bunch of us got together and we decided that this is the right time to organize a webinar on this very, very important topic. And so I'm Hema Somanathan from ISA Trivandrum, along with Axel Brockman and from NCBS, Vasuki Bellavadi from UAS Bangalore and GKVK, and Joe B. Joseph from University of Hyderabad and Parthiba Bosu from University of Calcutta. We've come up with about 8 speakers who agreed to give talks every Friday around the same time, 5 p.m. So we look forward to meeting you in subsequent weeks as well. So without taking too much time, I'll move on to introduce very briefly our speaker here. Alyssa is a pollination ecologist, and she works a lot on the pollination ecology of bats and also on bees. So today's talk, she's going to span bats as well as bees in her pollination research. Alyssa is from the University, Mahidol University in Thailand, and she's based there, and she does a lot of her work in the Thailand area, in forests in Thailand. And in India, we have a small bunch of researchers working on pollination, and this platform will also serve as an opportunity to collaborate with our Asian colleagues interested in pollination biology. So with that, Alyssa, I welcome you and I invite you to deliver your talk. So I'll just very briefly explain how we propose to go about with this. So about 15 minutes into Alyssa's talk, I'm going to act as a moderator for today, and I will look at whether there are any raised hands. So the button to raise hands is at the bottom of your screen. So anyone who has a question can raise your hand at any point of time, but reserve the questions when I will unmute your mic about 15 minutes into the talk, and whenever Alyssa thinks it's suitable to stop. And then you will have opportunities to ask questions, and then we'll do the same thing another 15 minutes later. So at 15 minutes and at 30 minutes, we'll have some questions. We can take some questions, and then we'll reserve the bulk of the questions for the end of the talk. So we'll try and stick to this format because we think that this is what will probably work better. So yeah, so thank you. And Alyssa, the stage is yours.
Okay, thank you. Is my microphone working okay? Can everyone hear me?
Yes, we can hear you.
Okay, great. Now let me share my presentation really quick. Can everyone see my slides now?
Yes.
Okay, great. So yes, I'm Alyssa. Thank you for the introduction and for the opportunity to talk about some of the work that I've been doing on pollinators in Thailand. So I'm sure with this audience, I don't have to spend too much time talking about the importance of pollinators. The majority of angiosperm species worldwide depend on animals, at least in part, for pollination. And this is especially true in the tropics. And there are a number of agricultural and economically and ecologically important plant species that rely on animals for pollination. So these pollinators are important not only for plant reproductive success, but also gene flow within and between populations, as well as plant community diversity. But I think a lot of people interested in pollination are initially interested in the flowers or the bees side of it. And my introduction to pollination was maybe a little backwards because I was initially interested in studying insect-eating bats. So these are some photos that I took in northeastern Thailand, and at around sunset every day, you can see these long ribbons of insectivorous bats flying out of the caves at night as they begin their foraging. And I've always thought bats were interesting. And after I completed my undergrad, I had the opportunity to work in a lab in southern Thailand that studied bats. And most of the students in that lab were working on insectivorous bats, but there was one grad student who was working on these nectar bats. So I grew up in a part of the US where we don't have nectar bats, we only have the insect-eating kind. And I did not even know that there were such things as bat pollinators. And as I worked with this grad student, I learned a lot about them and became really interested in them. And I'm guessing that maybe a lot of you don't have a lot of experience or don't know much about pollinating bats. And so I wanted to start with a brief introduction of bats in the Old World tropics. So most bat species worldwide and in the Old World tropics forage on insects, and there are many different families that are insectivorous. But there's also one family in the Old World tropics called the Pteropodidae, which are primarily or strictly nectarivorous or fruitivorous. So they only feed on fruits and flowers, and this is the family Pteropodidae. So just by looking at the images of the two groups, you probably notice a couple of striking differences. For the insectivorous bats, they have these tiny eyes and relatively large ears, while the pteropodid bats have relatively large eyes and small ears. And the reason for that is because insect-eating bats rely on echolocation to sense their environment and when they're foraging for prey. So because they're foraging at nighttime when it's dark, they don't rely on their vision. They rely on these large, well-developed ears, which helps them when they're echolocating. But the pteropodid bats are unique in that they are the only family of bats in the world that are not capable of true echolocation. So there are some species that have a primitive form of echolocation, but it's not the main sense that they rely on. These bats are mostly relying on their vision and their sense of smell. So because of that, they have really good eyesight that can see under dim lighting, and they have these relatively small ears because they don't really use them that much. And I think the most famous or the most well-known of the pteropodid bats are the flying foxes, as we see in this photo on the right. So even within the Pteropodidae, we see variation in morphology. This bat in the center is a nectar specialist, so it forages only on nectar, and accordingly, it has a really long muzzle and a long tongue, which help it extract nectar from flowers. And then here's an example of a pteropodid bat that mostly forages on fruit, but also occasionally drinks nectar. And so this bat has a much shorter snout and broad, powerful jaws and a very short tongue. So while I was learning about all of these nectar bat species, I was also learning about bat pollinated plants, which again, I previously did not know that there was such a thing. But in tropical regions, there are a number of plant species that are almost exclusively pollinated by bats. And these are the 6 taxa that I've been focusing on because they are all common in the diets of nectar bats in southern Thailand. You guys may be familiar with some of them, such as durian and banana. And even though these plants come from a broad range of families, they all share a number of traits that are common to plants in the bat pollination syndrome. So that means that plant species that are adapted to bat pollinators. And some of these traits include having flowers that bloom at night. So most of these species start blooming around dusk, and They will typically fall before morning, so that means they're completely dependent on nocturnal pollinators. Another trait is that they tend to be white or yellow or cream colored because these are the colors that are most visible at night when it's dark. They also tend to have relatively large flowers that are pretty sturdy. So nectar bats in the New World tropics can hover, and so sometimes you'll see delicate flowers in the New World tropics. But pteropodid bats like to land when they feed on nectar, so the flowers have to be large enough and sturdy enough to support the weight of a relatively large pollinator. And then also because these bats are relatively large and they need a lot of food, these flowers also produce a lot of nectar for their bat pollinators. So learning about this, I became really interested in the bat plant pollination interactions, and I decided to do my PhD on this study system. I did my PhD at the University of Maryland, but I was lucky enough to do all of my field work in southern Thailand. And one of the first questions that I had was, who is pollinating what? We know that we have all these pteropodid bat species and all of these bat-pollinated plant species. Is everyone pollinating everything, or are there species-specific interactions? We know from previous work that bats are definitely important pollinators because, for example, this is data from durian, and you can see that flowers that were visited by both bats and insects had a fruit set of around 5 to 10%, while those that were visited only by insects had 0% fruit set. And then we see similar findings for Parkia, where flowers that were visited by bats and insects had around 70% pollination success, and those that were visited only by insects had a mean pollination success of 0%. So we know these bats are important, but we still didn't have a good idea about which bat species were doing the pollination. So for example, here are some photos of banana inflorescences, and the three pictures have three different species of bat visiting. So on the left, we have Macroglossus sabrinus, in the middle is Eonectarus spalaei, on the right is Cynopterus sphinx. And even though all three species are visiting banana flowers, we don't know if they're all contributing equally to pollination. So in order to answer this question, we wanted to measure pollinator importance, which is defined as the product of visitation rate and pollen transfer efficiency. So for example, if we have a Parkia flower and we see that the gray bat species is visiting the flowers much more often than the brown species, but maybe it's not carrying as many pollen grains, and this brown species, if it's carrying a lot of pollen, it may end up being the more important pollinator, even though it visits the flowers less often. So in order to answer this question, I needed data on both the visitation rate and how many pollen grains these bats are carrying. So to do that, we caught the bats using mist nets. These are very similar to the mist nets used for catching birds. And it's basically like a fishing net, except that the line is very thin and very fine. So even in the daytime, it's difficult to see, and at nighttime, it's basically invisible. And we can set the mist nets in front of flowers that we know that the bats like. And then when they come to forage on the flowers, they end up getting tangled in the net, and then we can identify them as species and collect pollen from them. So one of the challenges of working with bat pollinators is that a lot of the flowers are quite tall. There are a lot of tree species that are bat pollinated. And so we were working with heights typically between 2 to 15 meters. And so we would use a slingshot to place guidelines in the canopy and then rig a pulley system so that we could pull the mist nets up to the appropriate height. But a common misconception when people would see me walking around with a slingshot was that I was going to shoot the bats. And so we always had to clarify that even if I were good enough to hit a moving target in the dark, which I'm not, we did not harm any bats during this research. So we caught the bats, and then we would also collect pollen from their fur. So in this photo, here is a bat that is carrying a ton of pollen. Usually they aren't carrying quite that much, but we can collect the pollen using glycerin gelatin, which you can see here in the syringe. And it's a little bit sticky, so when we run it through the bat's fur, it picks up pollen grains, which we can then mount on a slide and then use a regular compound microscope to identify and count the pollen grains. So this is a durian pollen grain, a parkia pollen grain, and a banana pollen grain. And you can see that they're all distinctive enough that it's pretty easy to identify them to species. So what we found was that there were some plant species where there was only a single important bat pollinator, such as was the case with Sabapentandra, Aroxylum indicum, and Parkia speciosa. For these three species, the only pollinator was this ES bat, which is Nectarivorous phyllae, which we can see here. And the reason for that was either because it was the only bat species that visited the flowers, such as in the case of Aroxylum, we really didn't catch any other species other than Nectarivorous phyllae. And then sometimes, such as with Parkia, we would catch other species, but they just weren't carrying a lot of Parkia pollen, and so we know that they aren't transferring pollen grains between flowers. One of the common names for this bat is the cave nectar bat, and so throughout this talk, I'm also going to be using the common name because we're going to be talking about the species a lot. So there were other plant species that we observed multiple important pollinators. For example, durian flowers. In addition to the cave nectar bat, we also observed Rosetta schleschenalti pollinating the flowers. For the banana flowers, we observed that the main pollinator was Macroglossus sabrinus, shown here on the right, and then to a lesser extent, the cave nectar bat and Macroglossus minimus. And then for Sonoratia mangroves, the main pollinator was Macroglossus minimus in the corner here, and then to a lesser extent, the cave nectar bat. So what these results were telling us is that the cave nectar bat is a very important pollinator for many different plant species, and it has a broad diet, so it can forage on lots of different plants. But the Macroglossus species are much more specialized in their feeding, and so, for example, Macroglossus sabrinus is a banana specialist, and Macroglossus minimus is a Sonoratia specialist. Now, when I was looking at these results, because the cave nectar bat is pollinating many different plant species, it led me to wonder, does that mean that this bat is moving pollen between all of these different plant species, or is there some mechanism preventing interspecific pollen transfer? And we hypothesized that given that these flowers have such different morphologies, that it's possible that the flowers are placing pollen on different areas of the bat. And this is beneficial to the plants because it can improve pollination efficiency. So for example, if a bat visits an Aroxylum flower and receives pollen on one area of its body, and then visits a Parkia flower where it receives pollen on a different area of its body, the Aroxylum pollen hasn't been disturbed yet, so that when it flies to another Aroxylum flower, this pollen can then be transferred to the second Aroxylum flower. And then same thing, if this bat were to go back to another Parkia flower, it would still have all this Parkia pollen that it could transfer to a new Parkia flower. So to test this hypothesis, we used a flight cage experiment where we would release bats into an empty room with a male flower and a female flower. Most of the plant species that we worked with are hermaphroditic, so we created functionally female flowers by removing the anthers from some of the flowers, but then we would leave the stigma so that we know that any pollen grains found on the stigma had to have come from the male flower. So here's an example of a cave nectar bat feeding on a male banana flower. And banana does actually have a male phase and a female phase. So this is a inflorescence in the male phase, and then we have an inflorescence in the female phase. You can see that it started to develop baby bananas. And we initially put a bag over the female flower so that the bat has to forage on the male flower first. And then when it's done foraging, we can pull a string to remove the bag from the female flower and then wait for the bat to come forage on the female flower. So this is an example of a conspecific trial where we used male and female flowers of the same species. We also looked at heterospecific trials. So in this case, we have a male parkia and a female banana. And in this case, the bat knows that there is a banana flower under this bag and spends the first few minutes trying to get at the banana flower before it eventually realizes that there is a parkia flower. So again, the bat feeds on the male flower. We remove the bag from the female flower. And in this case, we want to see if the bat is moving any pollen across flowers of different species. So after each trial, we would remove the stigmas from the female flowers and count the number of pollen grains that were transferred. And we would also collect pollen from 4 different locations on the bat's body: the crown of the head, the face, the chest, and the ventral side of one wing. And again, we use that same glycerin gelatin, mounted it on a slide, and then counted the number of pollen grains to see where these flowers are placing most of the pollen. What we found was that the bats transfer more pollen between flowers of the same species than between flowers of different species. So in this graph, the green points represent conspecific combinations and the black points represent heterospecific combinations. So for example, bats transferred around 200 pollen grains from male to female ceiba flowers, around 50 pollen grains from male to female dureo, and around 80 pollen grains from male to female banana. Whereas movement of pollen across species was much lower, typically less than 50 pollen grains. So, and these labels, the name on the top is the male flower and the name on the bottom is the female species that we used. Now, the reason why there was very little heterospecific pollen transfer is because these flowers were indeed placing pollen on different areas of the bat. In some cases, it was a very clear pattern. So for example, banana flowers place most of the pollen on the bat's face, parkia flowers place most of the pollen on the bat's chest, and this makes sense based on how the bats land on the flowers. So for banana flowers, the bat lands on the bract and then just sticks its face into these florets shown here in white and yellow. And for parkia, in this inflorescence, the fertile reproductive florets are at the bottom, and all of these florets at the top are actually non-fertile. They don't produce pollen. Their main function is to produce nectar. So when the bat lands on the flower to drink up the nectar, it ends up getting pollen along its chest. Now for other species, the pattern was less clear. For ceiba and dureo, we saw pretty broad pollen placement across most of the parts of the bat. And that was because these two species produce flowers in clusters, and the bat kind of lands on the entire cluster and ends up getting pollen everywhere. So based on this behavior, we would expect that there might be a lot of pollen transfer between ceiba and durian, except for the fact that they actually flower during different times of the year. So ceiba, at least in southern Thailand, flowers in January, durian flowers in March and April. And so because they have different flowering chronologies, there's never any overlap for bat pollinators. I wanted to mention one other species, Aroxylum indicum, which we weren't able to use in the flight cage experiment, but we hypothesized that based on floral morphology, this flower probably places pollen on the bat's on the crown of the bat's head and the face. And we were able to look at this hypothesis using bats that we caught from the wild that were foraging in the wild, not in our experimental room. And the results are pretty similar to what we found in the flight cage experiment. So one thing to point out is that we also looked at pollen accumulation over time. So on the x-axis, we have time from around 6 p.m. until midnight. And we can see that pollen does accumulate on the bats throughout the night. And similar to our previous results, the Ceiba and Durio flowers had pollen placement over all 4 areas of the bat. There was a lot of overlap. For Musa, most of the pollen was placed on the face, shown here in blue. And for Araxilum, as we hypothesized, most of the pollen placement was on the crown of the head and the face. And then again, for Parkia, most of the pollen was placed on the chest, shown here in green. So to wrap up the bat portion of this talk, these bats are really important pollinators, and in particular, the cave nectar bat pollinates a number of night blooming species, including agricultural crops like durian and parkia, and also ecologically important species like wild bananas and mangroves. And it's also important to note that there's not complete overlap in who is pollinating what. So for example, there's one species of mangrove, Sonoratia mangrove in Thailand, that is critically endangered. And we know that mangroves are a habitat that are disappearing worldwide, including in Thailand. And this Sonoratia mangrove depends on Macroglossus minimus for pollination. So knowing who the pollinators are can be helpful for conservation efforts of both the bats and the plants. And then finally, we found that even for the cave nectar bat that is visiting many different plant species, these bats are really efficient pollinators because most of the pollen transfer is between flowers of the same species. And that's because these flowers have different floral morphologies and place pollen on different areas of the bat.
Alyssa?
Yeah.
I think there is a raised hand. So maybe you can take a question now before you move on.
Sure.
Yeah. There are 2 raised hands. Bahiratan, maybe you can ask your question. Bahiratan? Okay, I think there was another raised hand. Sinu, do you want to ask your question now?
Yes, Hema, can you hear me?
Yes, we can hear you. Go ahead.
Yeah, Alyssa, thank you. So it's nice presentation. So I have 2 questions. Is the density of Ceiba flowers a reason why this this conspecificity is pretty high compared to the Parkia and Musa and other combinations? Because Ceiba has a large number of flowers. Is it a reason why the Ceiba conspecificity is pretty high?
I think that's part of it. And I think also, you know, there's variation in how many pollen grains that flowers produce. And we know that Ceiba does produce a lot of pollen. So
yes,
that's definitely part of it. And then, you know, for example, because the bats are landing on the cluster and getting pollen everywhere, it's very different from Musa, where they're just sticking their nose into the flower. Because really, Musa produces a lot of pollen as well, but it's just, it doesn't get spread all over the bat the way that Ceiba does.
All right, thank you. I have another curious question. You know, this is because now there is a corona and we had Nipah in Kerala and all. Is it sipping the nectar from the Musa flower still safe?
Is it do you know people who drink nectar from Musa?
Musa flowers, yeah, because we as kids used to sip, sip the nectar from the Musa flowers. So is it really safe now? What is your point on that?
My professional recommendation is to not sip the nectar.
Okay.
I don't have any data either way, but knowing that bats do like banana nectar, I would say it's probably better to not drink the nectar.
Okay.
And the other thing is that it depends also on when the flower blooms, right? So if you know that it's a freshly bloomed banana flower that, let's say it blooms, at 3 p.m. and we know there aren't any bat pollinators, I would say that's probably a safe flower to try drinking the nectar from. But if it's been open throughout the entire night, I would say find a different flower.
Yeah, thank you very much, Alyssa.
Yeah.
Alyssa, I had a question,
Sure.
if I may ask. So what was the best predictor of, you showed that there was a difference in the number of pollen grains that were carried by the different bat species. So what was the best predictor of the number of pollen grains? Was it body size or furriness or something else?
Are you referring to this graph in particular?
Uh-huh. Yeah.
Yeah. So it depends. The biggest predictor was the morphology of the flower and how the bat interacts with the flower. And then another part of it is how many pollen grains the flower produces, right? So, like with Parkia, we can see it with our own eyes whenever we catch bats at Parkia flowers, where their chest will just be covered in yellow pollen because that's where the bat is contacting the fertile flowers of the inflorescence. And, this other picture with Eroxylum, we can see very clearly that a lot of the pollen is getting deposited on the head. So the biggest factor is the shape of the flower and how the bat is interacting with the flower.
Okay. Yeah. Thank you. Yeah. So maybe you can continue your presentation and then we'll take questions at the end of your talk now.
Great. I just realized that I forgot to turn on my air conditioning, so I will be back in 20 seconds.
Sure. That's fine.
Okay, that should be more comfortable for me. Okay, so all of this work that I've done over the past decade was conducted in southern Thailand. And after I finished my PhD, I got a position working at Mahidol University, which is located in Bangkok. And the landscape of Bangkok is very different from the landscape of southern Thailand, and there aren't nearly as many bats. And so when I first moved, I started thinking about what are some projects that I can do locally? And I became interested in urban ecology. So urban ecology is a relatively new field because cities are a relatively new type of habitat, but it's one that's gaining a lot of attention, and we're starting to see a lot more work because we can learn a lot from urban ecology. So for example, we can understand how organisms are responding to human disturbance. We know that some species do very well in urban environments and other species don't. We can use the information to model future patterns because cities are one of the few habitat types that are going to continue to expand in the near future. So by studying these cities now, we can figure out what's going to happen to future cities that are continuing to grow. And we also know that most of the world's population growth is going to occur in tropical regions. And then finally, we can use this data to help shape urban planning. So we can figure out what factors that promote greater biodiversity within our cities, which is healthier not only for the organisms, but also for humans. We also know that urban ecology is important with respect to pollinators because pollinator populations are undergoing declines worldwide due to a number of different reasons. And one of the big reasons is habitat loss, and urbanization falls under that category. So let's take a look at an example of urbanization in Bangkok. This is a map of the city from 1945. We can see the Chao Phraya River running through the city in blue. And at this time, most of the development in the city was along the east side of the river. And now let's compare it to a map of Bangkok that is more current. Again, we can see the Chao Phraya River going through the city, but now the development has completely covered the entire area. And if we want to see the full extent of urbanization, we have to zoom way out and we can see how much Bangkok has grown over the past 75 years. And Bangkok is now home to over 10 million residents. So we know that this has a large impact on all species in the area, including pollinators. So one of the first things that I was interested in was, what are the spatial factors that influence pollinator abundance and diversity? Are there some factors that are actually beneficial to pollinators that promote their presence in cities? And then the second part was, what is the temporal variation in pollinator communities throughout the year? Because most studies on urban pollinators have been conducted in temperate areas where pollinators are not active during the winter months. But in tropical areas, we have flowers year-round and pollinators are active year-round. So I wanted to look at whether cities can be suitable habitat for pollinators across all seasons. So to address the first objective, we collected data from 52 green areas, and I define a green area as any location with abundant vegetation. And we ended up sampling 27 parks, 9 schools and universities, 8 temples, and 8 commercial or tourist areas, such as zoos or golf courses. And at each of these green areas, we conducted pollinator observations using 2 by 2 meter plots where we recorded all floral visitors and identified them to species where possible and recorded the number of individuals that we observed. And we also collected data on different spatial variables that we thought might have an impact on pollinator abundance and diversity. So these included the size of the green area, the type of the green area, which we classified as park, school, temple, or tourist area, and then plant diversity, floral abundance, and percent surrounding vegetation. And this last one, we used GIS methods to estimate the percentage of vegetation at different radii, all the way from 100 meter radii up to 1500 meter radii. And then we wanted to assess which of these factors is most important or has the biggest impact on pollinator abundance and diversity. So during those 3 months, we observed nearly 19,000 pollinators. So Bangkok definitely can support pollinators. These covered 38 taxa from 6 orders. But by far the most common pollinators that we observed were the tetragonulus stingless bees, which you can see in these top 2 videos. We observed them at every single green area that we visited. And then the next most common were 3 species of native honeybee. So we have Apis florea on the left, Apis dorsata on the right, and then Apis cerana was also very common. In addition to these taxa, we also observed a number of wasp species, sweat bees, carpenter bees, blue-banded bees, and 10 butterfly taxa. So the general breakdown, almost 99% were hymenopterans. We observed just over 1% butterflies and moths, and then very few individuals of flies, beetles, hemipterans, and then we also had one observation of a sunbird visiting a flower. So in terms of the factors that promoted pollinator diversity and abundance, there were two main ones, which were patch size and floral abundance. And as you can see from these graphs, the larger the green area and the more abundant the floral resources, the greater the pollinator diversity and abundance. And this makes sense because obviously the floral abundance is a reflection of the foraging resources that are available to the pollinators. But we also found a significant interaction between patch size and floral abundance. So in these graphs, you can see that the smallest green areas, shown in light gray, had very few pollinators even as floral abundance increased, whereas in the green areas that were above 1 hectare, we observed a positive correlation between the number of flowers and pollinator diversity and abundance. So what this was telling us was that there seems to be some minimum size limit where green areas that were less than 1 hectare are not very attractive or suitable foraging areas for pollinators, but anything greater than 1 hectare supports pollinators, and the more flowers that you have, the more pollinators that you're going to have. To address the second objective, we narrowed down to 9 focal parks because we needed to collect data every month. So these were the 9 parks that we focused on that were spaced throughout Bangkok. And we collected data for 12 consecutive months. So for each park, we would conduct pollinator observations the same as I described before. And we also collected data on the average monthly temperature, total monthly precipitation, and then patch size, floral abundance, and floral richness. And we found that pollinator abundance and richness remained very constant throughout the year. So the graph on the top shows temperature in red and precipitation in blue. Granted, because Bangkok is tropical, there's not much variation in temperature anyways. It ranged from around 26 to 30 degrees Celsius, but there was quite a bit of variation in precipitation ranging from 50 to 300 millimeters per month. And in spite of these fluctuations, the number of pollinators and the number of species of pollinators were pretty consistent across all months. So if we look at a breakdown of the pollinators that we observed, again, we find that the most common pollinators were tetragonulus stingless bees at around 60%. And then 30% of the individuals were our 3 species of native honeybee. And then the remaining 10% were other hymenopterans, hemipterans, lepidopterans, and dipterans. And in terms of the factors that promoted pollinator abundance and diversity, they were very similar to the first objective, where we found that floral abundance and plant richness were both positively correlated with pollinator abundance. And for pollinator richness, we found there were positive correlations with both floral species richness and precipitation as well. We also looked at pollination network, where in this figure, we have all of the pollinator species in yellow, all of the plant species in green, and the width of the line connecting them indicates the strength of the interaction. So again, we can see that stingless bees, shown here, visited the greatest diversity of plants and were the most important pollinators, followed by our 3 species of apis and Lasioglossum sweat bees. And then we also created pollination networks for each individual park during each of the 12 months, and we calculated different network metrics such as connectance and nestedness and the number of interacting partners and compartmentalization. And we found that these network metrics did not change across months. So that was indicating to us that these pollinator networks are stable across seasons, and there wasn't really much change at all. So in summary, the most common pollinators in Bangkok were the stingless bees, honeybees, sweat bees, butterflies, and all of the pollinators that we observed were native species to Thailand. And we ended up observing a total of 70 pollinator taxa from the two portions of that study. Just for comparison, a recent study in southern Thailand found 316 pollinator taxa in fruit orchards. So obviously, Bangkok is not supporting as many pollinator species as other more natural habitat types, but it's indicating to us that there are a large number of pollinator taxa that are okay with coexisting with humans in a highly disturbed environment. We also found that these pollinators are supported by a high diversity of cultivated plants, and the pollinators were equally happy at both the native and exotic plant species. So one thing that I didn't touch on in the results, but we did analyze whether pollinators had a preference for either native or exotic plant species, because over 50% of the plants that we observed were exotic species, and none of the pollinators exhibited a preference for either native or exotic. In terms of ways that we can promote pollinators in cities, the most important factors were the size of the green area, floral abundance, and floral richness. So again, if you're living in a city, plant lots of flowers and there will be pollinators. And another interesting aspect was that it's not restricted to just public parks. So we also tested whether pollinators exhibited a preference for parks over temples and schools and touristy areas, and there was no significant difference. So anywhere that has lots of vegetation and lots of flowers can be suitable foraging habitat for these pollinators. And then finally, we found that cities actually provide very stable habitats, and that's because humans are constantly landscaping, and we're constantly bringing in new flowering species. So a lot of the parks and other green areas, when one flowering species would, when the flowers would wilt, they would rotate that plant species out and then bring in a new species that was flowering. And so cities actually provide really convenient habitats for pollinators, and that there's a high abundance of flowers year round. So finally, I'd just like to thank the Indian Pollinator Initiative for inviting me to talk about my work, and everyone who's helped with all of the research. Most of the bat work was helped by colleagues from the University of Maryland and Prince of Songkhla University, and most of the urban bee research has been helped by colleagues and students from Mahidol University. And then, of course, my funding sources, and a big thank you to everyone here who decided to spend their Friday evening listening to me talk about pollinators. Thank you.
Thank you, Alyssa. There are a few questions here. And also, this is being streamed live on Google. So there are a few questions over on YouTube. So we'll take the questions here first. There is a question from Irene. Irene? And from Sinu.
Hello.
Yes, Irene.
Yeah, so I had a question. You had divided the green spaces like in terms of it being temples or parks and places like that. So is it just based on the physical places, or is it the type of flora or the type of plants that you see there?
Yeah, it was based strictly on how humans use the space, and it had nothing to do with the plant composition. So if it was a place of worship, we classified it as a temple. If it was a place of learning, we classified it as a school. And in general, there was a lot of overlap in the plant species that we observed, because humans tend to plant similar plant species throughout the city. So there was a lot of overlap, and there was a surprising amount of diversity in all of the habitat types that we observed.
Yeah, I wanted to ask this because there are sometimes there are specific plants that are in certain areas, like with the religious significance and all that. So did that play a certain role? So yeah, that was the question. Thank you so much. Thank you.
Yeah.
So there is a question from Sinu.
Yeah, thanks, Hema. Alyssa, I'm still with the bad presentation. I have two questions. One question is because the nocturnal flowers are very few compared to the diurnal flowers, or the one which is blooming in the day. And we have very few nocturnal visitors. What do you think on the reproductive fitness of the plants? Are the nocturnal plants more benefited than the the diurnal plants? So it's nothing about your specific presentation. I just want to clarify one of my hypotheses with this in my mind, because you have experience with the bat. Can you please tell me, are the nocturnal plants more reproductively benefited than the diurnal plants? That's my first question.
Mm-hmm. Yeah, I think there's going to be a lot of variation in both the nocturnal and the diurnal. I would agree with you that there's more potential for the nocturnal plants to receive fewer pollinator species, and it's more likely that it'll be it's more likely that there will be conspecific pollen transfer because there are just fewer pollinator species at night. But then there's also, as a plant becomes more specialized on a specific pollinator, there's also a greater risk that if that pollinator disappears or moves to a different area, then they could also run the risk of not getting pollinated, right? So I don't think I've ever read anything that has specifically compared pollination specificity between nocturnal and diurnal pollinators, but I think that your hypothesis about nocturnal pollinators being more species specific, I think that is probably true just because there are fewer nocturnal plants and fewer nocturnal pollinators.
Yes, thank you very much. Yeah, that is something which is in my mind and I want to work on. And the second question is regarding your urban work. Are the flowers sprayed by the pesticides? Have you counted that too?
That is a factor that we did not take into account, and it's a good question, and I don't have an answer to that. I know these plants are getting chemical fertilizer and they're getting artificial watering, and it's also likely that they're getting sprayed with, well, I won't say likely, I'll say it's possible that they're getting sprayed with chemical pesticides as well. Because actually, now I'm not sure, because if you think about it, like public parks where there's a lot of people going to exercise, I've never seen anyone spraying anything when I've been at the parks. And I'm not sure if they, if they do it when the parks are closed and not available to visitors, or if they're not doing it. That's a really good question. But based on the number of pollinators that we see, either if the plants are getting sprayed, the pollinators are still pretty abundant. I mean, even though we didn't see that grade of diversity, we did see very high abundance, especially with the stingless bees. And so I'll have to go back to the parks and ask whether pesticides are sprayed, and then comment in a future opportunity. Good question, though.
Yeah, Alyssa, the point I asked, because after all, they're all insects, and we know that insects are overcoming all kinds of pesticides. And I think that if they are pesticide affected too, I think the bees don't really mind. Parthiv may have a different viewpoint from mine, but because I'm an entomologist, I find managing insects is not that easy. So if you're seeing the same trend across the month and the same trend, it's not changing, I believe that they are not really affected if they are sprayed. But you told there are no evidences for that. Okay, thank you very much anyway, Alyssa.
So there's a question from Axel Brockman.
Yeah, hi Alyssa, nice talk. I was wondering, kind of in your plant-pollinator network, the honeybees, Florea was actually kind of like Florea and Serrana seem to pollinate more flowers than Dorsata, if I interpreted correctly. And actually, I would have thought naively that maybe Dorsata would be more important. Do you have any idea about that? Hello?
Alyssa?
So I think she needs to be allowed. I think Alyssa needs to be, the mic needs to be unmuted.
No, it's unmuted, but there's probably a small issue. We're trying to resolve that.
And one is,
we'll just have to wait a bit. So Alyssa is here, but is her mic muted? Alyssa? Yeah. We couldn't hear you briefly. But yes.
Can you guys hear me now? Sorry, I dropped out. If the previous participant could repeat your second question, or you were talking about how you're an entomologist and the effect of pesticides on insects.
What you asking me? So I was interested.
Axel, you can repeat your question, maybe.
Okay, good. So, because I'm a honeybee biologist, I was interested in kind of like the question, I mean, which of the three species might be most important for pollination? And naively, I would have thought of Dorsata playing a big role, just because they have larger colonies. And then actually your study shows that Florea is very important. And I wanted to know whether you have an idea why that is. I mean, kind of like maybe there are not many Apis dorsata colonies in Bangkok. Or the other thing was that Apis dorsata is nesting in aggregations. So I was not sure whether the distribution of Apis dorsata is basically homogeneous over the whole city. And kind of like you find places where you find more Apis dorsata and places where you don't find.
Yeah, I think you're exactly right when you were getting at their nesting habits. So I think because Dorsata does have these big colonies, and from my experience, they tend to build their hives pretty high up. So I would predict that there are fewer areas where Dorsata can build their hives in a city compared to Florea. But in terms of like how homogeneous they are throughout the city, I mean, we know that larger bees fly farther than smaller bees, and I would predict, I haven't analyzed the data, but just based on what I remember from collecting data, there weren't any areas that struck me as being particularly heavy concentrations of dorsata. And so I think because they can fly quite far, they're pretty homogeneous throughout the city. But that would be another interesting question to look into.
Yeah, and maybe we don't know anything about densities of flowea nests, right? Because hard to. Thank you very much.
Exactly.
There is a question from Binod Saradar. Binod?
Audible, ma'am?
Yes, we can hear you.
Audible?
Yes, you can go ahead.
Ma'am. In bat is the mammal. In nocturnal pollination, the moonlight and dark light, any effect? Light and dark light. Effect in nocturnal pollination for bat. Street light. Effect on nocturnal pollination. And summer and winter season. Effect on nocturnal pollination.
Okay, you're kind of breaking up, but I think your question is about the effect of street lights and artificial lights on bat pollinators, right?
Yes. And he also had another question, which was dark nights and bright nights.
Okay, thank you. Yeah, that is a good point. And we actually did a tiny study. Some colleagues of mine did a tiny study in China where they aimed different types of lights on flowers. So they used a white light, a red light, an infrared light, and a control that didn't have any light. And then they measured bat visitation rates to the flowers. And they found that bat visitation was higher to the control and the infrared light flowers compared to the white and red visible light. So we do know that light definitely has an impact on bat pollinators. But I think the bats do become acclimated to some extent to the lights, because a lot of the times we were catching bats in neighborhoods where there were street lights and there were lights from houses, and we still caught many bats. And so I think if it's something where, they're accustomed to the light being there every night, they are less affected by it. But if they have a choice where they can go to a darker area, they're more likely to visit the flowers in darker areas compared to well-exposed areas. Regarding your question about dark versus bright nights, that is something that I in particular haven't researched, but I know there have been studies that have found that bats are more cautious during full moon periods, and that can lead to fewer capture rates during bright nights, moonlit nights, but it's not something that I particularly have studied.
Thank you. There's a question from Kavya.
I'm sorry, can you repeat the question? Are they year-round residents or do they migrate? I know that dorsata does migrate, or they at least change their nesting site, and it's something that I haven't examined in Bangkok, but I would predict that their natural behaviors are still in effect, where once a hive gets too old, then they would abandon it and choose a new nesting area. So I haven't specifically studied that, but I assume that their behavior is still going to be pretty similar to wild roost or wild nesting dorsata. Yeah. Mm-hmm. Okay. So when we were analyzing the data, I didn't look at dorsata in particular. I looked at all apis bees just in order to have a higher sample size. But now that you're asking me, it makes me want to go back and reanalyze my data and look at it per species. But at the genus level, we saw a constant rate across months. So I'm sorry I can't answer your question specifically for dorsata.
There's a question. Devasahayam, can you ask your question, please? Okay, maybe we should move on to the next person. The Shiny has a question. Can you unmute your mic?
Hello, can you hear me?
Yes, we can. Yeah, go ahead.
Thank you, Alyssa, for your presentation. This is regarding the urban space studies. Were there any information or data to show any decline in the population of the pollinators in urban spaces over the years? Because all over we say, like, there's a pollinator decline, because of anthropogenic pressures. So from your study, were there any data which showed, like, these urban spaces do provide a habitat or refuge for pollinators?
Yeah, I wish I had long-term data to answer that question. I mean, ideally, we would have started collecting data 70 years ago when Bangkok was still tiny and observed the effect on urban pollinators. But, right now I only have 1 year of data. So you're going to have to ask me that question in about 10 years and I'll get back to you it's possible that because if floral resources are constant and these pollinator species are unfazed by human activity, it could be that we don't see any decline in pollinators over time compared to in natural habitats, right? But again, because urban ecology is such a new field and we're only just starting to study how pollinators are doing in urban habitats, I think it's still too early to answer that question, and we need to get a few more years of data first.
Thank you.
Partip, can you ask your question, please?
Yeah, thanks, Alyssa. Lovely presentation. Just one question: how polluted is Bangkok? And did you check out if it is polluted? Have you checked out the influence of pollutants, even suspended particulate matter on flower visitation, etc.?
We didn't have any methods to measure pollution in the city. I would say that Bangkok is fairly polluted. Every year we have issues where the air quality gets really bad, and so they recommend that everyone wears masks, which was really useful for this COVID pandemic because everyone in Bangkok already had masks because we use it for the poor air quality. And I'm not sure how big of an effect that has on the pollinators compared to other aspects of the city. For example, I think the the lack of natural habitat and the sheer number of humans moving around the city is likely going to have more of an impact on the pollinators than, for example, air pollution or other types of pollution. I haven't read any studies that have looked into that, but I think that would be something would be interesting to look at different tropical cities that have different levels of human density or different levels of pollutedness, and then we would be able to answer that question. But yeah, that's a good idea.
Thanks.
There's a question from Vasuki.
Yeah.
Yeah.
Alyssa, it was a nice presentation. I just wanted to ask whether there is any comparison with rural areas from the urban areas. Did you make any comparisons about the density or the diversity of the bees that you collected? Is there anything?
We did not collect data on bees in rural areas, but I know there have been other studies that have looked into that. And, in general, the cities support.
Because why I'm asking is in the rural areas, you may have more, or in the farmlands, you may have more application of pesticides than in the urban areas. And I think that might answer Dinu's question also, that probably the amount of pesticides used in the urban areas may be much less, and you may find higher diversity in urban areas compared to the farmlands or in the rural areas. So that's what, in fact, we did a small study, and that's the thing that we have come across around Bangalore. That's why I'm telling. Yeah.
That's an interesting finding, and I'm glad you shared that. I would say that, yeah, in that case, a big part of it probably has to do with the type of rural area. So, if it's intensively
like the distance from the urban area, you can take into consideration. Yeah. Thank you.
But yeah, I would say that the pollinator diversity in cities is promoted by the high diversity and the high abundance of plants. But then it's only going to work for pollinators that are unfazed by human activity. And we know that a lot of animal species don't like human activity, and like, for example, a lot of the ground nesting species, there's too much disturbance in urban environments to have them build their nest in the ground. Yeah.
Thank you.
Yeah.
There's a question from Pradipta Biswas. Can you unmute your mic?
Yeah, thank you. Lisa, it was a very, very nice talk. So I just wanted to know, have you made any comparison between the invasive plant species and the native plant species visitation rate or something like that? Because we all know that urban parks and all other green space, that are artificial plantation, maybe or not artificial, that maybe there's some ornamental invasive plants also we used to plant just to glorify or make that ornamentation. So have you seen anything particular in these issues?
Yep, we did a comparison to see whether pollinators preferred native or exotic plants, and there was no significant difference in visitation rates. So I think because the pollinators that can live in Bangkok, they tend to be generalist. They tend to have generalist foraging, like generalist diets and generalist nesting behaviors. They tend to be very adaptable. And so I think in that case, they were equally happy feeding on native and exotic plant species.
Okay. Thank you.
There's a question from Joby Joseph. Joby, you have to unmute your mic. Joby, we can't hear you. We can't hear you. Joby, we can't hear you. We can't hear you. There's an issue with your mic.
I'm not sure, maybe if you try unplugging your headphones and maybe trying the microphone on your laptop or your...
Yeah, I guess. But in the meanwhile, there are a few questions over YouTube, audience over YouTube. Maybe I can, go to some of those questions in the meanwhile. So someone was asking about, that it was noticed that bat species that foraged at lower heights were visiting a lesser number of plant species. Is that right? In your bat presentation?
Bats that forage at lower heights
also visited fewer species, yeah.
Also visited fewer species. I haven't looked at that in particular, but I do know from experience that all of the bat species that we study prefer foraging at flowers that are higher in the canopy. So for example, if you have a durian tree and there's a range of flowers between 2 to 10 meters, you're going to have higher success in catching the bats if you can put your mist nets up higher in the canopy. And I think it has to do with them just feeling safer higher up. So I don't have any data on the number of plant species versus foraging height, but I will say that from my experience, the bats do prefer foraging higher up.
Okay. There's another question, which is on stigmatic receptivity of bat-pollinated flowers and whether that is confined to the nighttime only.
I don't think it's been examined for all of the bat species, but I know it's been looked at in durian and Aroxylum. And for those two species, the stigmas are receptive throughout the night, and then the petals fall sometime between 1 to 4 a.m. And at that point, the stigma receptivity does start to decline as well. So these are definitely species that are adapted exclusively to nocturnal pollination.
Thank you. That was a question from Shankar Bhatt. And now there's another question from Salma Yaqub from Malaysia, who wants to know which was the most abundant wasp family collected in your parks?
Let's see if I have that data with me. I would, just based on memory, I think it's Campsomerus was the species that we observed the most often. We couldn't identify it to species, but genus Campsomerus was most frequently observed.
Yep. There's a question from Alexei Reshchikov. I hope I got that right. Are there any records of insects occasionally being attracted to bat-pollinated species? And if so, whether insectivorous bat species could interact with, I think, flowers occasionally? So that's the question.
Mm-hmm. Yep, we definitely observed. Actually, dorsata was the most common insect that we observed during the early evening, and I think for the Soneratia flowers, we occasionally observed some moths. So in terms of insect-eating bats, I think it's possible that they're contacting the pollen of flowers. I don't think it happens very commonly. I know there have been reports of it in the New World where they've reported that insect-eating bats like to come eat the insects that are visiting the flowers for nectar. But in my experience, there aren't enough insects at the flowers for it to be an attractive food source for the insect bats. But it could happen occasionally.
The question from Joby Joseph, whom we could not hear earlier. So he asks whether, when you compare counts across species, should we take into account their size / nectar / pollen capacity? One often finds only one sunbird or a few dorsata and many stingless bees on a plant, on a patch.
So is this referring to like pollination efficiency?
So I think in terms of abundance, maybe you had that graph with pollinator abundance. So should we take size into consideration here when you talk about abundance?
Yeah, I would say that goes back to when I was talking about pollinator importance. And one component is how frequently they're visiting the flowers, and another component is how much pollen they're transferring. So in that case, I don't think size of the pollinator is as important as how much pollen they're moving, because if it's a small stingless bee that is carrying lots of pollen and moving lots of pollen, then it could be a really effective pollinator. But that's one thing that we didn't collect data on that would have been useful is measuring how much pollen these pollinators are carrying. And I think that would be able to better answer your question. But I would say that size is not as important as how much pollen is being carried.
Okay, so maybe we can take a few more questions. There's one on coffee, but I'm not sure because I don't think you spoke about coffee here. But there's a question asking if the yield of coffee can increase with the help of pollination through honeybees in comparison to control plants.
I have heard about, I know a colleague who was interested in doing a study about this, about improving coffee yields through pollinators. I don't remember if it was honeybees. I actually think it was carpenter bees that he was interested in studying, but I know there's interest in it. So I think it's possible that coffee yields could be improved by either beekeeping or even just using more natural methods of promoting pollinator diversity in the farms.
Okay, so I think we've exhausted most questions. There were a few questions which were very similar, which I skipped through. So, and there are no more questions, raised hands here either on the platform. So thank you. Thanks a lot, Alyssa. This was wonderful. And you've set the tone very well for the rest of the series. We hope to see you in the remaining talks among the audience. Thank you very much. May I take the last question? Did you see nocturnal bees at your at flowers which were visited by bats at night?
We did occasionally see
Xylocopa in Thailand.
We have Xylocopa. I never observed them at bat pollinated flowers. The only bees that I observed at the bat pollinated flowers were Apis dorsata.
At night? At night, is it?
Yeah, it will. Yeah, so early evening. But even when it was completely dark at like 9 p.m., we would still occasionally come in to find Apis dorsata. And we actually really only observed them at the parkia flowers. I think because the parkia flowers produce so much nectar that it just pools up on the surface and it's a really easy food source. So I know that dorsata like parkia flowers.
Okay. So thank you so much. I think there are no further questions. So thanks very much for your fantastic talk and look forward to meeting you in person.
Thank you so much for the invitation to talk today.
And thanks to everyone who participated. Thank you.